Greetings, this is Dr. Jess. Welcome to part 4 of Ask Me Anything About Anything. This week we're going to talk about genes. That's a subject that everybody wants to know about, and I'm going to tell you the truth. I'm going to spread this information over 2 or 3 weeks because of the complexity of the subject. So today I'm going to tell you what they mean, how they fit into your healthcare, and we'll go on from there, okay? Well, genes and SNPs and pathways, oh my, it can get real complicated. What are genes? Well, let me tell you what you need to know. Genes control all hereditary characteristics of an organism that are transmitted from generation to generation. Oh, this is kind of obvious, right? And all the essential biochemical pathways responsible for the expression of the body functions are controlled by enzymes. Genes create enzymes. By themselves, genes don't do anything. They create enzymes. Why is it important for you to know that genes create enzymes? Well, enzymes are how your body processes different molecules to produce the products that you need for your bodily functions. So, I want you to know that the genes are unchanging. Genetic, or more accurately, enzymatic expression varies on a whole mess of different factors. So in the folate pathway you see on the left-hand side there, you see how the folate comes out of the body and becomes DHF, dihydrofolate, and then turns into tetrahydrofolate, and then turns into a bunch of fancy names ending in 5,10-methylene tetrahydrofolate, where MTHFR, methylene tetrahydrofolate reductase, reduces it to 5-methylfolate, which is what we use in our bodies. Fallacy number 1, you can change a gene. You cannot change a gene. The genes are there, they're there at birth, you don't change them. But you can influence the gene's expression. Let's take this whole thing a little bit at a time, one bite at a time. What do the SNPs, single nucleotide polymorphisms, what do they mean? They're an estimate of the enzyme's innate function. The genes, like MTHFR, creates an enzyme called methylene tetrahydrofolate reductase. Anytime you see an ASC on the end, that's an enzyme. But what do the polymorphisms or SNPs mean? Well, if it's green, minus minus, it is usually, think of it as a 8-lane highway. If it's heterozygous, plus minus are usually shown as yellow. It's heterozygous and it's got about approximately 60% enzyme function. And if it's homozygous, it's got about 10 to 20% enzyme function. Well, the heterozygous is like a 4-lane highway, and the homozygous is like a 2-lane highway. Let me throw something at you. If there's no traffic, does it matter at all whether the gene and resultant enzyme is normal, heterozygous, or homozygous? Not really. Things are going to function just nicely. But traffic will slow down the pathway's function. And it makes sense that given traffic, the homozygous or 2-lane highway will get blocked up faster than the heterozygous, and the heterozygous 4-lane highway will get blocked up faster than the 8-lane highway, or the normal. And what is the traffic? Bacteria, heavy metals, viruses, parasites, food allergens, candida, leaky gut syndrome, anything that creates oxidative stress. So if you have a lot of traffic, you're not going to get the products as quickly. And I have to tell you that when you look at something like MTHFR, there's not just 2 of them, because they always study the C6717 and the 1298C. When I say something fast, ignore it, okay? There are 50 of them, 50, and some programs will show you up to 15, which you'll be able to get a good estimate of the innate function of MTHFR. But back to this particular example, can you put enough traffic in an 8-lane highway to make it slow down and not give you the products? You think? Green or normal doesn't mean you're necessarily good to go. You can put enough traffic, enough oxidative stress into those normal looking pathways to slow it down. That's why you can't be guided just by the genes when you're evaluating healthcare. Fallacy number 2. If I have a SNP, I am sick. Well, I created a saying that I believe to be very true. The presence of a SNP does not mean you're ill, and the absence of a SNP does not mean you're well. Some programs present their genetic information in this manner. This is mthfrsupport.com, and by the way, this is mine, where you see lists of genes. The RS numbers on the left-hand side, that is the number that was given to the gene and particular variant by the Human Genome Project. And the middle, you'll see, let's say this first one on top, ABP/1/DAO. Okay, that's the gene's name, and then the particular variant. And then it gives you the present genetic construct and what the risk allele is. And of course, the easiest way to understand this is to look at the right-hand side with the yellow, green, and red indicators of whether we're having normal, heterozygous, or homozygous. Okay, this is a wonderful report, and it reads either the 23andMe or the Ancestry.com data. It's not very expensive, and it's consistently updated. The difficulty with this particular study is you really need to know a bit about what's going on, and the significance of the genes are not clear in the report, and you need somebody who really knows what they're doing to interpret it. My best suggestion is Strategene by Dr. Ben Lynch, because in the pathways that he shows, you'll see the gene, which you see on the top here. The green will have the cofactor, which is that vitamin and mineral combination that gets the enzyme to work. You'll also see whether it's slow, intermediate, fast, or complicated. Okay, and I'll tell you what that means in a little while. You'll see anything that decreases the activity in purple, and anything that increases or upregulates the activity in orange. So let's just briefly go over pathways. By the way, this is the Roche biochemical pathway chart. This is how I started learning about this way back when. And you can see it's intensely complicated. Okay, so what Dr. Ben has done with Strategene is kind of make it understandable, which is, he's really done a good job on it. So again, let's make this functional. When you see the bar on the bottom, the purple is slow, or think of it as homozygous. The yellow or intermediate, think of it as heterozygous. It's not exactly the way it is, but it's a good way to think about it. The fast is well fast, and complicated is like the girl telling you, it's complicated, okay? And that's for better interpretation, but I'll show you how things play out in a little bit. Okay, this is a pathway. This is my histamine pathway, okay? And I want you to notice where I've said on the top where you see B6 and B5, those are the cofactors for histamine decarboxylase and NAT2, okay? So you see what's going on here. Also, look on the left-hand side where you see DAO and the ALDH family. Okay, you see the products that these create, and the top one is hydrogen peroxide and ammonia, but primarily it creates an aldehyde. And then the aldehyde dehydrogenase, ALDH, reduces that to acetic acid, which is basically vinegar, so it can go out in the urine. Things like gut inflammation, metformin, alcohol acetaldehyde will slow down DAO, whereas in the LDH, increased reactive oxygen species, reactive nitrogen species, in other words, any kind of oxidative stress will lower the function of it. So here I have a low functioning ALDH with, if I have a bunch of oxidative stress, it can make it even worse, but I need zinc and B3. What you can do with this is you can't change the ALDH, but what you can do is supply it with the cofactors and do work to get rid of the oxidative stress, okay? The upregulating factors, let's look at the ALDH at the bottom, are things like zinc, increased hydrogen sulfide, vitamin B3, and those things that will downregulate, I've already said. Think of upregulation and downregulation this way. You can have a heterozygous SNP and let's say a normal amount of traffic or a slightly increased amount of traffic, and have downregulating factors that we showed you before, and that's going to make it a functional homozygous. So the traffic that's in heterozygous, okay, that makes it barely work, okay, is then going to be almost pushed into a two-lane highway, sort of like having construction on a highway. If you've ever driven like that, you know everything slows down real good, okay, because of the down-regulating factors like the construction people. So it's a little easier to understand it like that. Okay, people, this is what really grinds my gears. MTHFR. Too many people have made MTHFR a deity or a devil, and it's not. It's not waiting in the tall grass to get you. By itself, MTHFR doesn't cause all the nasty things that it's been assigned to it. The reason that you see that stuff is because in the very beginning, all they did was study MTHFR2 variants, and they thought it would be a way to understand higher homocysteine. That didn't work out so well. So a lot of people with any kind of problems would look at their MTHFR, and if there were any polymorphisms, they would assign that particular symptom or condition to MTHFR. That is not what happens. Okay, MTHFR turns 5,10-methylene tetrahydrofolate into 5-methylfolate, which is what you use for folate in your body, and it's about 1/3 of the methylation pathway. It can't be considered by itself. So the polymorphisms of SNP are only relevant in relation to their function within a particular pathway, the presence or absence of cofactors and coenzymes that will help determine their overall function, the downregulating and upregulating factors, and the relationship of the presenting complaints and relevant testing. You can't look at a genetic study and say, this is what's wrong with you. Okay, the genetic study points you in directions, and if you have a polymorphic pathway, which we're going to go over next week, then you say, gee, I got a lot of oxidative stress, and maybe it's going to express in a certain way because that pathway doesn't look like it would do well under an oxidative stress load. And that's the truth. So next week, we're going to talk about how to interpret genetic pathways and what they mean to you in your health, okay? And what to look out for when you're commissioning an interpretation of the pathways, okay? Doing it yourself is tough, okay, to get the real information. And I want you to be careful. Okay, people, I want you to be careful. There are programs out there that will lead you directly or somewhat indirectly to either their products or certain products. This is inherently a bias, and I suggest you don't trust it, as the information is going to be skewed towards the pharmaceutical or nutraceutical protocols. There are programs out there that read your genetics and either with a basic amount or minimal amount of information or no information tell you what you should be taking. Those are inherently not good, trust me. Okay. Okay, please subscribe to my YouTube channel and tell your friends, because over the next few weeks we're going to get into the nitty-gritty of things and information that you'd really like to know. If you have questions, you can either email office@drjessonline.com or email me directly at jess@drjessonline.com. If you'd like to know if I can help you, please go to my website and schedule a 30-minute get acquainted session. It is free, and we can discuss your issues, and I can let you know in no uncertain terms whether I can help you or not. Why would you want to treat with me? Well, I'm very well educated. I've been a healthcare provider for 5 decades now, and I'm the doctor that will give you the time and empathy you need. As always, if no one told you that they love you today, don't forget Dr. Jess does. And I'll see you next week.